All right, today we're going on a pretty incredible journey. We're going to trace the story of heredity, how we understand it all, and we're starting from a simple garden and taking it all the way to the code that's literally inside every single one of our cells. So yeah, let's jump right in. I mean, you've probably wondered this, right? Why do I have my dad's nose but my mom's laugh? It's that classic question. Why do we look like our family? How does some traits get passed on while others just seem to pop up randomly? Well, the answers to those questions, that's the foundation of genetics, right there. And get this, the story doesn't kick off in some fancy, high-tech lab like you might think. Nope. It starts in a quiet monastery garden with a monk, a whole lot of patience, and believe it or not, thousands and thousands of pea plants. This is where the blueprint of life first started to give up its secrets. So let's meet our main character, Gregor Mendel. You've probably heard him called the father of modern genetics, and for good reason. See, he was the first person who didn't just look at inheritance. He systematically counted and analyzed everything. He basically took this big biological mystery and turned it into a math problem that he could solve. Yeah, this here really gets to the heart of his breakthrough. Before Mendel, the common idea was that traits just kind of blended, like mixing red and white paint to get pink. But Mendel, with his thousands of pea plants, he said, no, that's not it. He figured out that inheritance is controlled by these separate little packets of information, these factors that get passed down. Today, of course, we call them genes. And that one idea, it literally changed everything. So the big question is, how did he actually crack this code? Well, Mendel came up with a set of rules, we call them laws now, that perfectly described how these factors of his behaved. These laws are like the absolute bedrock of genetics. But before we can really dig into them, we need to get on the same page with a couple of key terms. Okay, first up, allele. It sounds technical, but it's pretty simple. Just think of a gene as a category, right? Let's say flower color. The alleles are just the different options in that category. So you could have a purple allele or a white allele. Super simple. You get one of these from each parent for every gene. And this is a really important distinction, genotype versus phenotype. The genotype, that's the genetic code itself, the actual combination of alleles you've got on the inside. The phenotype is what you can actually see. It's the physical trait that shows up, like the purple flowers. So to put it simply, genotype is the instruction manual, phenotype is the finished product. Okay, now that we've got those terms down, let's tackle Mendel's first two laws. First, the law of dominance. This just means that some alleles are kind of bossy. They're dominant. If you have one, that's the trait you're going to see. The other one, the recessive allele, it's still there, but it gets hidden. Then there's the law of segregation. This one explains that when it's time to make sperm or eggs, you only pass on one of your two alleles for any given trait. They segregate or separate. So how can we predict what's going to happen? We use this cool little tool called a Punnett square. It's actually pretty easy. Let's walk through it. See, we put the alleles from one parent up top, a big P and a little p. Then we put the other parent's alleles down the side. Same thing, big P, little p. Now you just fill in the grid like a multiplication table. Big P plus big P gives us PP. Big P and little p, that's PP. You do the same thing for the bottom row. And now look at what we've got. 3 of these boxes, PP and the 2 PPs, are going to be purple flowers because that dominant P is there. Only the one little PP box gives us white flowers. And boom, there it is, that perfect 3-to-1 ratio of purple to white that Mendel kept seeing in his garden. And then there's his final rule, the law of independent assortment. This takes things a step further. Basically, it says that the gene for one thing, like flower color, has absolutely nothing to do with the gene for something else, like how tall the plant is. They're inherited independently. It's kind of like shuffling 2 separate decks of cards at the same time. What you get in one deck doesn't affect what you get in the other. Okay, so Mendel's work was just brilliant, right? But for decades after he published it, there was this massive nagging question. He talked about these abstract factors, but nobody knew what they actually were. I mean, where in the cell were they? The next part of our story is all about finding the physical home for those instructions. This was the huge missing piece of the puzzle. If these things were real, they had to be physical. But what were they? And where the heck were they hiding? And here it is. This is the answer. Scientists finally discovered that Mendel's factors, what we now call genes, are physically located on these little thread-like things inside the nucleus of our cells, and those things are called chromosomes. This was the moment. This is when the idea became a real, physical thing you could see under a microscope. This whole breakthrough got a formal name, the chromosomal theory of inheritance. And you can't overstate how huge this was. It was like building a bridge between two separate islands. On one side, you had Mendel's genetics, all math and patterns. On the other, you had cell biology, people looking at physical things in cells. Suddenly, scientists realized that the way chromosomes physically moved and separated during cell division, it was a perfect match. It physically explained the exact mathematical patterns Mendel had found. Everything just clicked into place. But, biology is never that simple, right? Just when you think you have the rules figured out, you find exceptions. And as scientists dug deeper, they found some really interesting new layers that built on Mendel's original work. One of the biggest exceptions to Mendel's rules is something called gene linkage. Remember that law of independent assortment, the whole separate decks of cards thing? Well, it turns out that's not always true. If genes are physically located really close to each other on the same chromosome, they don't assort independently. They tend to get inherited together, kind of like two friends who always travel in the same car. And then there's the whole topic of how sex is determined, which is just fascinating. For us humans, we're all familiar with the XY system, right? XX is female, XY is male. But nature has way more tricks up its sleeve. Take a look at this chart. In birds, like chickens, they use a totally different system called ZW, and it's flipped. In their world, the female has the two different chromosomes, ZW, and the male has two of the same, ZZ. Kind of wild, right? So we've established we have this incredible genetic blueprint, this instruction manual for life. But what happens if there's a typo? What if something in the code gets changed? Well, that's where we get into the world of mutations and genetic disorders. A mutation, at its core, is just a change in that genetic material. And these changes can happen on totally different scales. It can be super small, like a single letter in the DNA code gets swapped out. That's what causes sickle cell anemia. Or it can be a massive change, affecting huge chunks of a chromosome, or even a whole extra chromosome. And when the change is about the number of chromosomes, we have a specific name for it, aneuploidy. So instead of having the normal 2 copies of a chromosome, a person might end up with an extra one or maybe be missing one. This kind of imbalance in the genetic recipe can have really significant effects, and it's often the cause of various genetic disorders. This chart really helps visualize what we're talking about. A typical human cell has 46 chromosomes, right? 23 pairs. But look here. Someone with Down syndrome has 47. That's because they have an extra copy of chromosome 21, which is why it's also called trisomy 21. Or take Klinefelter syndrome. That's also 47 chromosomes, but in this case, it's because of an extra X chromosome, leading to that XXY genotype. All right, let's pull it all together and recap the big takeaways from our story. First, inheritance is not just a random mess. It actually follows predictable mathematical rules, which we have Gregor Mendel to thank for. Second, those mysterious factors he talked about, they're real, they're genes, and they live on our chromosomes. That's the chromosomal theory. And finally, we've seen that any changes to this blueprint, mutations, are the ultimate source of all genetic variation, and sometimes they can cause disease. So think about where we've been. We started in a quiet garden, just counting pea plants, and we followed that thread of knowledge all the way down to the code of life itself. We went from counting peas to counting chromosomes. It's an incredible story. And it leaves us with a pretty profound question, doesn't it? Now that we understand the rules of the game so well, what happens next? What does it mean for our future now that we're developing the tools to not just read the blueprint, but to actually start rewriting it?